Hello everyone and welcome back to our channel Biodiversity and Conservation. This is Lakshmi Rao from Ambika Prasad Research Foundation, Odisha, India. And today we are going to study about some cell signaling receptors like the enzyme linked receptors, of which we covered the receptor tyrosine kinases. We are going to cover another two types of enzyme linked receptors, that is tyrosine kinase associated receptors and receptor serotonin kinases, along with what is the effect of nitrogen oxide in the signaling process, and how chemotaxis is seen in bacteria and also quorum sensing. So let's start. So the second enzyme linked receptor is tyrosine kinases associated receptors, and these type of receptors contain N-terminal extracellular ligand binding domains, single transmembrane alpha helices, and C-terminal cytosolic domains. But the only difference from RTKs, that is receptor tyrosine kinases, is that the cytosolic domain of tyrosine kinases associated receptors are devoid of any known catalytic activity. Instead, these receptors function in association with non-receptor cytosolic tyrosine kinases, which are activated as a result of ligand binding. And a variety of receptor classes belong to this category, including the receptors of antigen and interleukins of lymphocytes, integrins and receptors for various cytokines, and some polypeptide hormones. The largest and most diverse class of tyrosine kinases associated receptors is the cytokine receptors. These class of receptors include the receptors for most cytokines like interleukin 2, erythropoietin, and some polypeptide hormones like growth hormone, prolactin, whose cytosolic domains are closely associated with a member of family cytosolic tyrosine kinases, that is the Janus kinases, which we know as JAKs. JAKs phosphorylate and activate the gene regulatory proteins called STATs, that is signal transducers and activators of transcription. And the STAT proteins are located in the cytosol, and after activation migrate into the nucleus and regulate the gene transcription. So here is this pathway of JAK-STAT signaling. So the JAK kinases, from the characteristic presence of two kinase domains in each molecule, several members of the family are known. There is JAK1, JAK2, JAK3, and type 2. Each of these receptors, each of these enzymes are associated with a specific set of cytokine receptors, and the JAK kinases are tyrosine kinases whose major substrates are transcription factors called STATs, and there are several types of STATs, each phosphorylated by a particular set of JAK kinases, and all the STAT proteins contain an N-terminal SH2 domain that binds to a phosphotyrosine in the receptor cytosolic domain, a central DNA binding domain, and a C-terminal domain with a critical tyrosine residue. Once a STAT is bound to the receptor, the C-terminal tyrosine is phosphorylated by an associated JAK kinase, and the STATs then dissociate from the receptors and form either homodimers or heterodimers. The Basis for dimerization is a reciprocal interaction between an SH2 domain in one subunit and a phosphorylated tyrosine in the other subunit. And the stat dimers move into the nucleus where they bind to specific enhancer sequences controlling the target genes and helps in the gene expression. Now, the next kind of receptor is serotonin kinase receptors. It is another enzyme-linked receptor. So, these serotonin receptors transduce signals for the TGF beta family, and these receptors are actually single pass transmembrane proteins with a serotonin kinase domain on the cytosolic end. There are mainly two classes of these receptors, that is type 1 and type 2. And the TGF beta superfamily consists of a large number of structurally related secretory dimeric proteins. The superfamily includes the TGF betas themselves, the activins, and bone morphogenetic proteins, that is BMPs. The BMPs constitute the largest family, and each member of the TGF beta superfamily binds to a characteristic combination of type 1 and type 2 receptors, both of which are required for signaling. And typically, the ligand first binds to and activates a type 2 receptor homodimer, which recruits and phosphorylates and activates the type 1 receptor homodimer, forming an active tetrameric receptor complex. Once activated, the receptor complexes use a strategy or for rapidly relaying the signal to the nucleus that is very similar to the Jack-Stat strategy. The type 1 receptor directly binds and phosphorylates a latent gene regulatory protein of the SMAD family. The activation of SMADs causes their translocation from the cytoplasm to the nucleus where they function to control the gene expression. Here, SMAD2 or 3 and SMAD4, they all migrate to the nucleus and recruit the other gene regulatory proteins and activates the transcription of specific target genes. And after that, we can see the targeted gene transcription or gene expression. So the next type of signaling that we often see is nitric oxide signaling. So here, nitric oxide is a paracrine signaling molecule. Like the steroid hormones, nitric oxide is able to diffuse directly across the plasma membrane of its target cells. And the nitric oxide is synthesized from the amino acid arginine by the enzyme NOS, that is nitric oxide synthase. Once synthesized, nitric oxide diffuses out of the cell and can act locally to affect nearby cells. In mammals, one well characterized example of nitric oxide action is the relaxed smooth muscle. This causes the dilation of the blood vessels. The autonomic nerves in the blood vessel walls release acetylcholine, a neurotransmitter. The acetylcholine acts on endothelial cells that line the interior of the vessel to stimulate enosynthesis. Then the nitric oxide then diffuses to the neighboring smooth muscle cells in the walls of blood vessels where it interacts with iron bound to the active site of the enzyme guanylyl cyclase. This increases the enzymatic activity, resulting in the synthesis of the secondary messenger cyclic GMP, which induces the muscle cell relaxation and blood vessel dilation. Thus, guanylyl cyclase acts both as an intracellular receptor for nitric oxide and as an intracellular signaling protein. Many types of nerve cells directly produce nitric oxide to signal to their neighbors. For example, nitric oxide released by autonomic nerves uh, causes the local blood vessel dilation that is responsible for phenyl erection. And nitric oxide is also produced by activated macrophages and neutrophils to help them to kill the invading microorganisms. Now, this is the diagram showing the chemotaxis in bacteria. So the movement of organisms in specific directions in response to the chemical stimulus is called chemotaxis. Chemotaxis in bacteria depends on a signaling pathway that terminates at the flagellar motor. Bacterial flagellar motion is rotatory in nature, and the membrane proteins MOT A and MOT B, along with the fleet G, create a proton channel that drives the rotation of the flagellum. And the rotation of flagella can be clockwise or counterclockwise. When the flagella rotate clockwise, forward motion ceases and the cells tumble, but the counterclockwise rotations of flagella impart the forward motion. In the absence of a chemical gradient, bacteria such as E. coli moves in a random fashion that includes the forward motion, where the cell is swimming forward and tumbles when the cell stops. Following a tumble, the direction of the next forward motion is random. However, if a gradient of a chemical attractant is present, these random movements become biased. As the bacterium senses that it is moving toward higher concentration of the attractant, forward motions become longer and tumbles less frequent. If the bacteria is sensing a repellent, the same general mechanism applies, although in this case it is the decrease in the concentration of the repellent that promotes the forward motion. So, bacterial chemotaxis is mediated by transmembrane receptors and a phosphorylation relay system. It depends on two-component signaling pathway activated by histidine kinase associated receptors. The chemotaxis receptors are methylated during adaptation and so are also called methyl accepting chemotaxis proteins MCPs. The phosphorylation relay system enables the chemotaxis receptors to control the flagellar motor. In E. coli, the binding of a repellent increases the activity of the receptor, which binds JW, that is an adapter protein, and JA, a sensor protein, which acts as a histidine kinase, thereby stimulating JA to phosphorylate itself on histidine. JA quickly transfers its covalently bound high energy phosphate directed to aspartate of JY, which is a response regulator to generate JY phosphate. The phosphorylated JY dissociates from the receptor, diffuses through the cytosol, and binds to the flagellar motor and causes it to rotate clockwise, resulting in tumbling. The phosphorylated state of JY remains only for a few seconds. Protein chain Z accelerates the dephosphorylation of JY phosphate, thereby inactivating it. The binding of an attractant has the opposite effect. It inactivates the receptor and therefore decreases the phosphorylation of JA and JY, which results in the counterclockwise flagellar rotation and forward motion. Um, The response to an increase in the concentration of an attractant or repellent is only transient, even if the higher level of ligand is maintained, as the bacteria desensitize or adapt to the increased stimulus. So now this is quorum sensing. Here the term quorum means quorum sensing describes bacterial communication phenomena that allows the bacteria to communicate using secreted signal molecules to assess their population density. This process actually enables a population of bacteria to collectively regulate gene expression and thereby behavior. In quorum sensing, bacteria access their population density by detecting the concentration of a particular signal molecule termed autoinducer, which is correlated with the cell density. Quorum sensing is the regulation of gene expression in response to fluctuations in cell population density, and bacteria that use quorum sensing constantly produce and secrete certain signaling molecules called autoinducers. These bacteria also have a receptor for the autoinducer. When the inducer binds to the receptor, it activates the transcription of a set of genes, including those responsible for the synthesis of the autoinducer itself. The concentration of the autoinducer in the surrounding medium depends on the cell population density. As the bacterial population grows, the concentration of the autoinducer in the surroundings increases, causing more autoinducer molecules to be synthesized. Um, Both gram-positive and gram-negative bacteria use quorum sensing communication circuits to regulate a diverse array of physiological activities. Um, in general, gram-negative bacteria use N-acyl L-homoserine lactones, that is AHLS, as autoinducers, and gram-positive bacteria use oligopeptides to communicate. So, the first incidence of this phenomena uh, came to light with the discovery of luminescence produced by the marine bacteria Vibrio fischeri. These bacteria, when free living in seawater, that is at low cell density, are non-luminescent. However, when grown to high cell densities, um, uh, they show bioluminescence with blue-green light. This bacteria commonly forms symbiotic relationships with fishes and squid species. They carry a specialized organ called light organ, in which bacteria luminescent appearance in dark environments is due to the maintenance of the high density of Vibrio fishery population in the light organ. In the marine environment, the bacteria only luminesces when colonizing the light organs and do not emit light when in the free living state. So the synthesis of lipophilic AHL is catalyzed by an enzyme called ALH synthase, the product of LUX1 gene, and the LUX1 gene is subject to positive autoregulation, that is, transcription of LUX1 increases as AHL accumulates in the cell. This is accomplished through a transcriptional activator LuxR, which is active only when it binds AHL. Thus, without AHL activated LuxR, the LuxI gene will be transcribed only at basal level. AHL freely diffuses out of the cell and accumulates in the environment. When the cell density increases, the concentration of AHL also increases. So the greater concentration of AHL flows back into the cell and activates high level transcription of LuxI and other genes whose products are needed for bioluminescence. Um, so this is all from me today. Thank you.